Today? Good. So, apparently I'm not getting an introduction, so I'll introduce myself. So that's good.
No, he doesn't.
So, David, I did this introduction the same way Tom does. We need, the picture of Crosse with and all these things that slide across the screen saying that he teaches pretty much everything in the world of mycology this guy teaches.
Yeah.
So for those of you who don't know the rock star of mycology, you haven't been mushrooming for very long. And I guess the way I'll introduce you today, Tom, is my first meeting with Tom. And that was back when I was a kid, back when I was young still. I think it was 2008, 2009, or 98, 99, yeah, way back. See? So what happened to you? I was walking out of the woods, it was my first big foray, and it was still the worst foray I've ever been to. It was extremely dry Maine woods, and I had this dried up Armillaria, when the honey mushrooms in my hand. And I see this, back then it was bleached blonde haired guy walking towards me. I was like, I've seen this guy before, since I knew of his website. And I said, that's Tom Volk. And so imagine for me, I was not a normal kid. So seeing Tom Volk in the woods was like stumbling upon a baseball star at the baseball field. So he looks at the Armillaria in my hand, he's like, yeah, it was just like typical Tom deadpan. That's Armillaria Melia. And I was like, oh, that's probably Armillaria Melia. And I was like, thanks, Tom. Before you, we used to be able to call them all Armillaria Melia.
Yeah.
And okay, so should have known then that would be some smart ass and kick me out of the mycology world, but you didn't, so you're stuck with me now. But the work that he's done, and I don't know, after the passing of Orson Miller, I think Orson's torch was passed to Tom as kind of the professional mycologist who really interacts with the amateur community and the wonderful tying link that many of you in the room, professional mycologists in the room also have, but Tom does such a great job of that. And so Tom Volk from the University of Wisconsin with Crosse, welcome to California.
Okay, wow, that was worth it. So thanks for coming to my talk, and I'm going to talk about cryptic species. And so these are the hidden gems of mycology, as I've said here, and these are species that are, we assume we know what they are, but we really don't. And so we're going to find out about a few of those today. And here's my NOAA slide to tell you that I teach at the University of Wisconsin-La Crosse, and we're on the Mississippi River in western Wisconsin. Here's the Mississippi River. My office is right here. So I'm a couple miles from the river, actually. We have about 9,000 students at UW-La Crosse, and about 1,200 of those are biology and microbiology majors. So we have really a big chunk of the school. I teach a lot of courses in mycology. Every fall we teach a general mycology course. Every spring I teach medical mycology, so these are fungi that infect people in various ways, so like yeast infections and ringworm and worse things you get out here in California like coccidioidomycosis, valley fever, lots of things like that. I teach plant microbe interactions where we look at how plants interact with fungi and bacteria and nematodes and viruses and what have you. Teach advanced mycology once in a while. Food and industrial mycology we teach once in a while, so how to exploit fungal life cycles to do what we want them to do and make stuff for us, like citric acid and vitamins and the steroids and the birth control pills and all kinds of things like that. I teach plant biology once in a while, and every spring I teach organismal biology, the second semester freshman course. They don't know what to make of me. And every fall I teach Latin and Greek for scientists, which is pretty fun. And so I'm thinking maybe someday I should do like a workshop on Latin and Greek at a Foray. So maybe I'll do that someday. Okay, so I'm going to talk today about different species. So we're going to talk about genera first. So I'm going to ask you a question, is this agaricus? Okay, so is this agaricus? So who votes yes, this is agaricus? Okay. Okay, so how about this? Who votes that this is agaricus? Okay. So who votes that this is agaricus? So who votes that this is agaricus? Okay, these are all agaricus. According to Elias Magnus Fries, he called every fungus, the father of modern mycology, he called every guild mushroom agaricus. Okay, and so you're willing to accept that now we have divided all of these into different groups. There's about 150 to 200 genera of agaricus now. Okay, so this is a natural progression. By learning about these fungi, we know more about them and we can change their names and we can figure out what's going on. Likewise, the same progression is going to take place with the species. Okay, so we wanted to call everything Armillaria melia. Now we have more data that says, no, that's not true. So if you're willing to accept that not all of those are agaricus, you must be willing to accept that Armillaria melia is not a single species. Okay, so that's the thesis of my argument here. Okay, let's see if I can convince you. Okay, so there's a lot of diversity in fungi. About 75,000 species of fungi have names, and depending on how you count things, there's an estimated 1.5 million species of fungi by several different kinds of estimates. So that means that about 95% of fungal species are unknown to science. Okay, so every year my students go out and they find something we don't know. And so, and you saw from Noah's and Christian's talk yesterday that probably a lot of the things on the East Coast are misnamed, and a lot of the things on the West Coast are named from things from the East Coast and from Europe, and those names aren't right either. So we'll see there's a lot of new species. But it seems like we hear about species diversity and we hear about the tropical rainforest, and like this is some sort of great place where everything exists. And it may be a great place where everything exists, but we don't know what we have here, as it turns out. And I'm going to give you some examples of that in a minute. There's a lot of different hosts in the tropics. If you go into a forest here, you're going to see, 3-4 species of trees in one area. In the tropics, there's one tree of this species here, and the next tree of the same species might be 100 meters away, and there's another 100 species in between them. So there's much more diversity of trees in the tropics, so there's much more diversity of the fungi that associate with them naturally. Okay? But here in the temperate zone, there are a lot of species, and they're right here under our noses, and we didn't see them because we assumed a lot of things that turn out not to be true. Okay? So these cryptic species are, you might complain that there's new names for everything. Okay? You didn't complain when we came up with a new name for agaricus at the beginning. Right? So it's a matter of just learning the things that are there. Okay? So we have lots of new data, and I'm going to show you different ways of finding these new species. Okay? So the ones we're going to talk about today, these are all species we thought we knew very well. So Armillaria melia, the honey mushroom, we assumed that we knew that very well at one time. There was one species, and that was it. Okay? There's not. Laetiporus sulfurous, the chicken of the woods, we assumed that we knew everything about that. There was one species, and that was it. And that's not true. What about morels? We were using the names Morchella esculenta, Morchella elata, Morchella semilibra, Morchella conica in North America. None of those species exist in North America. Not a single one. Okay? And for many years, we used the name Chanterelle siberius for all of our chanterelles. There is no Chanterelle siberius in North America. Okay, so we're going to talk about all of those different species and the various ways of distinguishing between the species. Okay, so in the last 20 to 30 years, we've made significant strides in species identification. Before that, most species identification was based totally on morphology, some ecology, but mostly micromorphology as well as some macromorphology thrown in. But now we have lots of new tools. We have molecular biology techniques. We have DNA sequencing preceded by PCR. And so we've got a lot of different ways to use those data to figure out what we have. Okay, so my first question is, what is a species? So this is a fundamental concept that you learn when you're a very small child because you have a see and say, and you pull the cord, it goes around and around, and the cow says moo. It's a cow. It's easy. It's not a chicken. It's not a duck. Right? So you're used to putting animals, these barnyard animals, and then eventually any young boy, at least worth his salt, knows the names of the dinosaurs, and all of those are different species, very clearly different species. But there's no training like that for mushrooms, right? There's no see and say for mushrooms, right? And so there's different concepts that you have to use for fungi than for other kinds of organisms. And it turns out these concepts we're going to talk about are also useful for other kinds of organisms. So I'm going to ask you again at the end, what is a species, and see if you know any more than you know now. Okay? So there are different ways of looking at species and determining what species are. And one of them that we look at a lot are these three are morphological species. So it looks different, it's a different species. The second one, and this is kind of your Reader's Digest slide, and I'm giving you the New York Times version in the next several slides. Okay, so this is, the biological species says that if two organisms can mate and form fertile offspring, they're the same species. Okay? The evolutionary species looks at the DNA, for the most part, and says that if two species have a common ancestor at some certain level that's sort of undetermined, they are the same species. Okay? So there's very, very different ways of looking at it. Okay? So you're very familiar with the morphological species concept. This is, you look at something and you figure out what it is. So whether this includes macromorphology and micromorphology both, some people are even hesitant to use microscopes. But these are things that you would use to distinguish between species, distinguish between genera and everything else as well. Okay? So this includes looking at the fruiting body for the most part, because that's what we see. We don't see the mycelium that's underground, and most of those mycelia all look the same anyway. They're very difficult even for an expert to distinguish between. And so we use the morphological species concept based on these morphology. So we decide what's important. We decide that having gills is more important than holds things together better than something that has pores. Okay? This gets thrown out when we look at gilled boletes. We look at boletes that have gills, but they're more closely related to the ones that have pores. And there's lots of things like that that already are giving us an indication that maybe our eyes are deceiving us for a large portion of the time. Okay? There are lots of very good mycologists who relied totally on micromorphology and got it. They saw that there were differences between russula and agarics, and they said, no, russulas are not related to the other agarics. There's something else. Okay? And no one believed them. Okay? But now we do, for other reasons we'll talk about. Okay? The biological species concept seems pretty straightforward. If you can take two organisms from two populations and they're compatible with one another, they can form fertile offspring, then they're the same biological species. So in the biological species concept, we do not consider whether the mating actually takes place. So according to the biological species concept, lions and tigers can interbreed and form ligers and tigons, depending on who the mother and who the father is, and they're fertile. So lions and tigers are the same biological species. Now, they never see one another. They're on different continents. But if you stick them together in a cage or a bar with soft lighting and a couple ferns and some wine, they'll mate. Okay? So you can't always measure whether that mating takes place. We can do that with fungi. So we can go collect spores from a fungus from Europe and mate it with the spores from a fungus from North America. And if they're compatible, they form a dikaryon, they produce fertile offspring, they're the same species. Okay? And so Ron Peterson has done a lot of this. He's traveled all over the world, collected spores from what we're calling, for example, Pinellus stypticus. And they're all the same. They mate. And when you bring them back and put them in a petri dish. However, Pinellus stypticus in North American populations, have a gene that codes for the glowing, the luminescence of the fruiting body. But that gene does not exist in the European population. Arguing against there being gene flow from the North American population to the European population. Doesn't prove anything, but it argues against it. Okay? So the biological species concept gets a little hazy when we're talking about whether the mating actually takes place. We don't have enough data. That's really the only piece of data we have that says maybe it does, maybe it doesn't. Okay? So geography has to be related to this because species can diverge by being on different continents. You know, they were all together in the Pangea and they broke apart. The species were free to change from one another. There was no selection for the mating genes that would allow them to mate with one another. And so we see this sometimes with animals when the flow of a river changes and ones on one side of the river become incompatible with the ones on the other side of the river over geologic time. But that's sort of a moot point because spores go back and forth across the river with fungi. And so there's bigger differences than that that have to be there for fungi to separate. Some of the difference may have to do with time. And so if one of them sporulates in the spring and one of them sporulates in the fall, they're probably not going to see each other, depending on how they grow. And so the same thing if on certain trees, they have both male cones and female cones, and they're fertile at different times. So they avoid pollinating one another. Okay, so there's all kinds of things like that that contribute to the biological species. So it's much more complex than what I'm giving you in this short slide here. Okay, are you still with me? Okay. Okay, now let's talk about the evolutionary species. So the evolutionary species says that any two individuals that can be traced to a common ancestral lineage are the same species. Okay, so we say that a species is monophyletic, and that says that monophyletic means that thing in the first sentence. Paraphyletic means that they contain members from one group, but they're excluding some of them. And polyphyletic means that they don't have a common ancestor. And so there's just a little terminology that's just for, in case you've heard those words before. The problem is, where do you draw the line? How many generations do you have to go back to find the common ancestor? And where do you draw the line? So this is an important problem in distinguishing between species. Where do you draw the line to say these are totally different from one, these are different species? Okay? Ideally, you want to use all three of these species concepts together. Okay, so for example, when molecular biology came about, you could see hundreds of papers that just published DNA sequences, and they published phylogenetic trees, and that was it. And they made no comments about the morphology and no comments about the biological species. You can't publish a paper like that anymore, okay? Because you have to have something else besides your molecular biology that will back up everything else, that will be backed up. Okay, so we're going to look at some examples of this and you'll understand how it works. Okay, so with the evolutionary species, the morphological species and the biological species must have something to do with it. There has to be something about it. If the mating type genes are free to change, the morphology and other things are free to change as well, the other thing, the DNA. Okay, so we're going to talk about the first one we're going to talk about is really the first one that sort of was the big important one, which was Armillaria mellia. Okay, so for many years, first we argued about whether to call it Armillaria or Armillariella, and Armillaria won for arcane reasons that I won't tell you about. But here are some examples of what used to be called Armillaria mellia. And so Armillaria mellia is considered a single species, worldwide in distribution, that was enormously variable in its morphology, enormously variable in its pathology, so whether it was a pathogen to trees or whether it just ate dead wood. So that was it was hard to believe that this was a single species that was doing all these different things. So even when we look at these species now, they don't look anything alike. You know, they had attached white gills and an annulus, and that's about all they have in common. Okay? And a lot of them had rhizomorphs, and some of them didn't. So these black shoestring things. So in the 1970s, a group from Scandinavia said they didn't believe this, that it was a single species. And they started looking at collecting single spores from fruiting bodies and putting them on petri dishes together and see what happened when they got stuck together. Okay? And so here is an example of a compatible relationship. So these are capable of mating with one another. So we have fungus here and one you guys put in here. And when we put them together, actually they're like this, and you can't see the line between them. They form a dikaryon, indicated by, this is an indication that they've mated, and they form a different kind of morphology that's happening here. Okay, and Armillaria is a little more difficult to fruit, but people have taken these and then fruited them from that. Okay, here's an incompatible relationship. Here's two isolates, and when you put them together, there's this kind of no man's land in between them where they fight one another off. They don't recognize that they're of the same species. They don't have the same mating type genes that are compatible with one another, and they fight each other off. Okay? And so that's all you see in an incompatible mating. And so Kari Koronen and his colleagues, and anybody remember where he's from? I can't remember whether it's Finland or Norway. Finland, thank you. And so he did these matings and collected spores from all over Europe and came up with 5 or 6 biological species from Europe. So in the 80s or so, Bob Ulrich's student, who is Jim Anderson, thank you. My names are bad today for some reason. It's the time zone change I blame it on. So Jim Anderson started doing this at the University of Vermont and did this with the North American species. And he found that there were about 10 biological species in North America. And some of them seem to be the same as the ones from Europe. Okay? And so he came up with these North American biological species. They weren't all named at the time. They certainly weren't named at all when he found them. They were all called Armillaria mellia. But gradually they were given names that you see here. The most recently named one is Armillaria altimontana, which was named last month. It took people that long to find it. So this is a weird thing. We had the mycelium from the wild, but we didn't know what it looked like in a fruiting body. So nobody knew how to collect it, and nobody did it systemically. And it's a very limited range species, seems to occur in northern Idaho and a few other places, but that's the main places in Idaho where you find it. The species before that was Armillaria nabsnona, which I named with Hal Birdsall from Olympic Peninsula in Washington. And so that occurs all down the west coast here on hardwoods. And so some of these are very well distributed and some of them are not very well distributed. So there is Armillaria mellia still exists. It's very common in the east, and it's also relatively common in Southern California, like south of sort of Bakersfield is where you might find Armillaria mellia. And I'm not convinced that's the same species. So more work needs to be done on that. One name you might not recognize is Armillaria solitipes, and in the past this has been called Armillaria stoyi. And so it turns out Armillaria solitipes is an older name for it. And Hal Bertzel and I wrote a paper showing this. It was when we were doing our book on the different species that had been in Armillaria at one time. We looked at the type specimen and saw that it looked like Armillaria, North American biological species 1. And so Peck's name predates the French name Armillaria stoyi by several decades, and so it should be the older name for it anyway. So the forest pathologists were really mad, like sending hate mail and things like that, because we were changing the name of a common pathogenic species that everybody knew and nobody wanted to change. They published papers and journals that said, this is an outrage and we're not going to change and we don't care about the rules of nomenclature. And then there was a formal effort to conserve the name Armillaria stoyi over solitipes. And in the last couple months, there's new data presented at meetings that says Armillaria solitipes from North America is not the same as Armillaria stoyi from Europe. And so the real name in North America is Armillaria solitipes. And so that's the way it goes. So we win. So Armillaria solitipes is a name from Colorado. It's the only Armillaria species in Colorado. So that's what you get. The data that came, let's just skip this slide. At the time when we were working on these biological species and wanted to work on species 9, the only good kind of information that was available to us was RFLP, a restriction fragment length polymorphism, where we cut the DNA with these enzymes that come from bacteria. They're recognizing foreign DNA, and they cut the DNA at very specific sites. And so if you get a sequence of DNA and you cut it with these restriction enzymes, and you run it on one of these gels, which separates the fragments based on size, you can get different patterns based on the species. And so here we got in Armillaria stoyi, we got this band. Armillaria sinapina had a couple different ones. Armillaria gallica had a couple. Armillaria sapistipes was slightly different, and Armillaria nubsnona had a couple different patterns. And so this was our first sort of molecular indication that these were different species. Okay? And then later people did DNA sequencing, which I want to talk about in the context of other species. Okay, does that all make sense? Good with me so far? Okay, how about Laetiporus sulfurous? Okay, everybody knows what that is. It's the chicken of woods. They're all the same. But when I give talks on my favorite mushrooms to eat on the East Coast, and I make the audience make a list of theirs, every single person has Laetiporus in their top 10. When I give the same talk on the West Coast, like 2% of the people have Laetiporus in their top 10. So that's the first indication something's amiss. The tastes aren't that different. I mean, I know that California is different from other places, but not that much. And so here's a Laetiporus. I like this email that I'll read to you. Lady wrote to me, multitudes of sulfur shelves are growing in my garden, apparently from the underground remains of 2 large oaks which were removed and mulched 10 years ago. Is there any way to eliminate the mushroom growth since it's all over my lawn? It's near my swing set and disturbs the young children who play there. It seems as soon as I remove one cluster, another one appears. Help. I felt so bad for her. So my reply, of course, was that she should educate the children about what they are and maybe they can learn something about nature. And she did not like that, apparently. I'd never heard from her again. So here's the chicken of the woods. In case you were wondering, one of my former students brought his daughter into the woods with the chicken, and I mean, they found it there. That's where they found the kid and the chicken. And so when we look at Laetiporus, there probably are now at least 6 species in North America, maybe 7. There's probably 2 in California. There's 4 or so in the east. There's probably a 5th one that's still undescribed. That's a different problem. All of them cause a brown rot. So they eat the cellulose of the wood and leave the lignin behind. So they're reasonably primitive, probably. Most of them cause a brown butt rot, which I hope is not as painful to the tree as it sounds. And a couple of at least one species causes a brown root rot. And so it grows from the ground in a rosette instead of from the side of the tree. So that's this Laetiporus cincinnatus. This is Laetiporus conifericula. This is my only picture from Alaska for Kate. And so here are the Laetiporus species. They're actually really easy to tell apart morphologically and geographically. So if you know the host, you know where you're standing. You can look it up on your phone if you have to. And you know that it's a Laetiporus, you can figure out what it is. Okay? And so here's Laetiporus conifericula is here, growing on conifer, west coast. Here is this is Laetiporus, sorry, this is Laetiporus sulfurous. This is Laetiporus conifericula from the Olympic Peninsula. This is Laetiporus gilbertsonii, variety pallida, that grows on the Gulf Coast of the US. And Laetiporus gilbertsonii, variety gilbertsonii, which grows on eucalyptus. So they're the same species growing on eucalyptus on the east, on the west coast, and on live oak mostly on the Gulf Coast. So we're still confused about that. They don't look the same, but they're not distinguishable by mating, which I'll show you in a minute, or by the ITS region. So it needs a little more work. Here is Laetiporus cincinnatus. So it is orange on the top, white underneath. It's the most delicious by far of all of the Laetiporus species. It's soft all the time. You can eat the whole thing all the time, not just the very edge. And it grows in huge rosettes on the ground. It's a root rot. Okay. This is Laetiporus persicinus, which you may have seen from the southeast US. It's not a Laetiporus. Throw it out. Okay. And this is an undescribed species that we've seen from the Midwest, and we've also had collections from Connecticut, so it's probably more widespread than that. And it is not very edible. It is tough. It turns black when you cook it, and it's bitter. Okay. And for a while, we thought it might be a sterile hybrid between Laetiporus sulfurous and Laetiporus cincinnatus. That would have made a great story, but it's wrong. Oh well. Okay. And so here's the mating studies in Laetiporus. So here's compatible ones. You stick the 2 individuals together on the plate, and they make this zone that's different from the zone of either one separately. So in this one, there were 2 spores, 2 mycelia placed together here, and then the same one placed here and the same one placed on the outside here. So you can see both of those started to look like. And so this is compatible, this is incompatible. So they're just kind of finding one another and kind of growing over the top of one another. So these would be the same species, these are different species. Okay? It's a little trickier than the Armillaria because there's no clamp connections in Laetiporus, so you can't look at the hyphae to actually see what's happening. So there's some limitations to this, but over time this is corroborated with all of our other kinds of data. This is an RFLP that when we were first looking at these, we distinguished between the species. And again, we looked at how they were cut by different enzymes, and you got these different patterns that were in different species. And then it's like a Chinese menu. You pick one from column A, one from column B, one from column C, and you put them all together, and you get a certain species. Again, I won't show you those data. It's not that exciting. Okay, and so this was sort of primitive data until we got DNA sequencing. And so Dan Lindner and Mark Bannock at the Forest Products Lab, where I used to work, who I worked with, started looking at the ITS region of the DNA. And so this is a place, it's called the internal transcribed spacer. That's not that important, but it's part of the DNA that forms the ribosome. Okay, and so you want to find an area that's different between species and not too similar between species. So over early experimentation with different genes and different DNA sequences, this is the one that people decided worked for fungi, because it does for the most part. And when you can only have one sequence, you can compare your ITS sequence with everybody else's sequence that was published in an online database called GenBank. Okay? So DNA has this advantage. So when you looked at morphology, we had, 10 characters or 15 characters at the most that we're using to separate. And when we're looking at DNA studies, every single DNA base is a character. So if you have a sequence of 500 DNA base pairs, you have 500 different characters to base your answer on when you compare the two. Okay? And so it's very, very powerful statistically compared to the statistics you can get out of just 10 or 15 different kinds of morphology. Okay? And before computers, this was impossible. So now we have computers, and they're rather sophisticated computer programs that like 3 people in the world understand that tell us how this works. Okay, so here's the large subunit sequence for the nuclear large subunit. So maybe a good T-shirt. And here's the ITS region for Laetiporus cincinnatus. So you got all those base pairs in there, all ready to go, and you can stick that, copy that, paste it into the sequences on GenBank online, and in a minute it comes back with all the matches for that sequence. Okay? And then you can use the things you match with to construct a phylogenetic tree, or you make your own DNA sequences from all of those different species. And this is what Lindner and Bannock got in their paper. It's not important that you see these things over here, but you can see that the different species are clearly separated from one another. And so what this did was give us the confidence to separate these species from one another. So we had another set of data besides the morphology, the geography, and everything else in the mating to say, yes, we're right. This is another set of data that's very powerful that gave us the confidence to put these other names on these other specimens. So it turns out we were right. Looking at our morphology and looking at our geography, we separated the species based on that before we did any kind of DNA studies. Okay, so here is our weird one down here, the one I said is an undescribed species that doesn't taste good. In this gene, it clusters out with Laetiporus sulfurous, and in the other gene, it's off by itself. So we're still debating about how much difference has to be enough difference to make it a different species. Certainly the morphology is different, and the mating, actually the spores don't germinate, which is one of our reasons we thought it might be a sterile hybrid. So, it's unclear what's going on. But in any case, using lots of different DNA regions, we can separate the species from one another and have the confidence to accept that our morphology and our geography and our biological species were correct. Okay? And so these clearly are different. Very, very clear which species they are based on this morphology. And so this is one time it worked. With the Armillaria, it's still really difficult to distinguish between the species because although the mating type genes have changed, the DNA has changed in many of them, the morphology hasn't changed very much. Not really. And so the Armillaria species are still pretty variable within the species, and they're still hard to tell from one another. With practice, you can do it, but you need just a little more practice, I think. All right, so how about Morels? So I'm not going to give you the big, long, hairy spiel about this, but just give you some idea of what happened with Morels. When was it, 2009, Kerry O'Donnell and a bunch of people published a paper from USD lab in Peoria that showed a phylogeny of Morels that was different, and showed that there was nothing that matched Morchella esculenta from Europe. Nothing that matched, nobody knows what Morchella crassipes is. And Morchella deliciosa, no one knew what that was either. There's no type specimens to match with it, and there's nothing in Europe that described like that. The black Morels, we had the names Morchella elata and Morchella conica from Europe, and we had the Peck name from New York, Morchella angusticeps, for that, and that's often used. And the half-free morels were called Morchella semilibera all over. So it turns out there are none of these, except for Morchella angusticeps, exist in North America. So nothing matches the DNA, or the mating is. I did some of the mating when I was in grad school, and it's sort of not very definitive. It's not as clean as the other species that we tried to look at. Okay, and so here's our paper, taxonomic revision of the true morels, Morchella in Canada and the United States. There's 11 authors. Not all of them got along. And so that's another very, very long story that I don't want to ever recount. But in any case, this is the phylogenetic tree. They do group very clearly into the black morels and the yellow morels. The semilibera goes with the black morels. So it's just one that the black morels all have a space between the hymenophore, where the spores are born, and the cap. Between the cap and the stem, there's a little gap in there underneath it. And in Morchella semilibera, that gap is exaggerated, so it goes halfway up. Okay, so it turns out there is a yellow black morel because of this gap, and that should have given us, that's better indication than the color. And so all the ones that are here in gray are newly described species. Many of the other ones are European species, and the European species are in different clades than the North American ones. So they're sister species, but they're very different from one another in terms of their DNA. Okay, and so here are pictures of all of them in our paper. This is from our paper. It's available online at mycologia.org. Just Google my name and it'll come up, and it's actually free. We got free access for everybody for it. And so here's Rufobrunia, diminutiva, virginiana. These 2 and 3 grow on tulip poplar in the southeast. Morchella esculentoides is the most common morel species in North America, the classic yellow, but it occurs out here as well. Morchella prava, Morchella cryptica, also on southeastern species. Morchella tomentosa after the burn, and Morchella frustrata that grows in bark mulch, and one of the Morchella semilibera look like Morchella punctipes, and the other one Morchella populophylla. The black morels include these that are all from the west. The only name we could reuse was Morchella angusticeps, and that was sketchy, and we had to do some taxonomic rigmarole to actually be able to use that Peck name, and it seems restricted to the east, so you don't have Morchella angusticeps in the west at all. But instead you have these 5 other black morel species in the west. And so it's unlikely that we have all of them, although Michael Kuo, who headed up this study, has been collecting morels from all over the country for 20 years. So he's got a pretty good indication. But we did find this one, Morchella snyderi. It's the only known from the type specimen that was collected by Snyder at the University of Washington in the early 1900s. So whether it still exists in nature is questionable, and we don't know what it really is. And we have specimens of it, but it doesn't seem to be very common. So it's likely there are other species like this that aren't very common that have evolved as well, that are in different places. Okay, so you can see there's some big differences in here, like this Morchella importuna has this very ladder-like structure on it. Some people separate the green one. If you go in the burns, there are green ones and pink ones and yellow ones. They're all in different species. So you can't tell the species by the green one. There are different species that are green. So it's environmentally determined and not genetically determined. And so I don't know how to distinguish between all the species. I've never seen all of them live, and so good luck. And so there are differences in the flavor, and so if you're only interested in for gastronomic purposes, you might want to still know the species. Go question?
Can we still eat them?
You can eat all of them. As long as you cook them well, you can eat all of them. Okay, but some of them, I think the burn morels are tasteless to me. I don't think they have any flavor at all compared to the eastern ones. Part of that, I think, comes from the bacteria and the yeast that live in the eastern ones that are not there in the fire ones because they got burned off in the fire. And so the only ones I've been able to culture and not be totally contaminated have been western morels from the fires. And the eastern ones all have bacteria and yeast associated with them. So I think that's what's going on. That's the only data I have, though. Okay? Mycologia.org. Yeah. And so you can find that. Just email me and I'll give you the website. Okay, so that's all I'm going to talk about morels. You can work it out. The other thing I should mention is that during this time, a French pharmacist named Philippe Clouz published a paper based on pictures he saw on Michael Crose's website and named probably 6 of our North American species without doing any kind of DNA work and he had specimens, but some of them were frozen, some were dried. And so people from this country sent him things. And he published the paper based totally on, this one looks like the picture on Michael Crose's website, so I'm going to call it that. And so we don't know, it's not totally resolved yet. People are, now the sequencing is being done by people from Europe and trying to figure out whether any of those really are the same and are they valid names. And so that remains to be seen. So this has happened just in the last several months. Okay? And so the jury's still out. You can call them whatever you want, and I don't care. Okay. Last thing I'll talk about are chanterelles. And until very recently, Chanterelle cibarius, considered the most widespread species in North America, was found under conifer and hardwood. It's enormously variable in its flavor, enormously variable in its morphology. Does any of this sound familiar? And so people started actually looking at the chanterelles. So there were some species that we considered distinct. So Chanterelle cinnabarinus was always just considered distinct and different. Here's one of Ron Pastorina's. Are you here, Ron? Oh, good. So he won't know that I used it. I already asked. So this is Chanterelle minor. It's a tiny little thing found in the east. Chanterelle subalbidus was white, and so that was easy. Cantharus latericius didn't have any ridges on it from the southeast, and it was always considered a different species. There was something called Cantharellus persicinus. I still don't know what that is. It's sort of peach-colored from Georgia, of all places. Huh? And so even early in 1976, Ron Peterson published a really big paper, Ron Peterson from the University of Tennessee, a long paper that said Cantharellus cibarius and Cantharellus cibarius in Europe and in North America were different from one another. Just based on the morphology. He was a very careful morphologist at the time, and he knew what he was doing looking at these things. And he said that across the 2 continents, the name Cantharellus cibarius was being applied to 8 to 10 taxa and probably many more. So this is very prophetic in 1979 when he said this, before there was any kind of easy molecular techniques to be able to be employed. With Cantharellus, we have the problem of that the fungus is an obligate mycorrhiza. So that means that it has to have the living tree to grow. That means that we can't force it to mate and culture because we can't culture it. Okay, so we're losing one of the legs of that triad of identification. We don't have a biological species concept in here. Okay, and so in the 1990s, Toby Feivelman, who some of you must know, and Bill Sabula, who many of you probably know or knew, were the first to really look at molecular techniques in using with chanterelles. And so they sequenced a region called the ITS region, and they found out that this is that same region we're using in some of the other genes, some of the other species. They found that the ITS region was really variable in size. So this was different than any of the other kinds of species. The ITS region was enormously long. It turns out it has a huge insert of DNA in it and became enormously long because of that. And so when they presented this data at an early meeting, no one believed them because they were coming from a lab that was not known for its DNA sequencing and not known for any of its other kind of work like that. And so everyone in the audience thought they were wrong because they had this enormously long ITS region and no one had ever seen anything like this before. But they were right. They were right. Okay? And so from that work, they started to look at the southeastern species, and this is the one they really the only one they described was Chanterelle tabernensis. It's a small thing about this size. I've been to this type of locality. It's at a tavern on the NASA headquarters where Bill Sabula worked in Stennis, Mississippi. And so there it is, and it looks way different than the other ones we've looked at. Just to continue on with the southern chanterelles, Bart Buchan and what's her first name? Valerie, thank you. I'm bad with names today. They described some new species from the southern U.S., Cantharellus tenuithrix and Cantharellus altipes. Okay? And so these are different using DNA techniques and using morphology from the other chanterelles. And although this looks kind of similar to Cantharellus tabernensis, it's not the same at all. And different colors and different kind of patterns of different hosts. Okay. In the West, things were a little bit ahead of that. A name was resurrected from the literature for the most common chanterelle on the West Coast, I think, is Cantharellus formosus. It's rather common and looks different from the others as well. In 2003, Cantharellus cascadensis was delimited by Susie Dunham and her colleagues, and here's that. That's mostly in the Cascades in Oregon, Washington. I think it gets down here into California. And then we had Cantharellus californicus by David Aurora and Susie Dunham did this paper. This is a weird big mushroom. Sometimes they call them mud puppies because they're in the mud. I've only seen them live once. I went on a foray with the Los Angeles Mycological Society and the San Diego Society joint foray somewhere in the mountains between there. And we were under live oak, and the duff for the live oak was this thick. Okay? And I was with one person whose name I can't remember who it is now. I tried all day to remember. It was Steve Pencall. And he would go and he'd say, "There's one right there." And he would see that one of those leaves was turned slightly differently. And he would dig down and find a chanterelle that was this big. So dig down a foot down into the duff and find that. Okay? And nobody else found anything. But he knew how to go into that duff and find. Have you been able to find him with him? Oh, is that right? Okay. So they're apparently worth it, and it grows in other places as well where it's easier to find, but this is a little bit more difficult. Okay, so another species was described by Redhead, Norville, and Donnell called Cantharellus siberius variety roseocanus. And this was described from Oregon, and not much was known about its distribution. It's a beautiful thing. It's very yellowish, orange underneath, and sort of rainbowy. That they recommended the common name rainbow chanterelle. And so you've got this. So now we know more about this distribution of this thing. Scott Redhead recently raised this to species level, Cantharellus roseocanus, which we were about to do anyway. So I'm going to show you some data that corroborates his information. So I had a grad student, Matt Foltz, who worked on chanterelles, and this was and the year he started working on it was the best chanterelle year ever. So this rarely happens to me where those two things coincide, that both of these things are going to happen together. So here's some of his collection of chanterelles. And when I was with him in the woods, I found two chanterelles, and I asked, "Are these different?" And that was the start of his thesis. Okay, so is this one that's yellow underneath different from the one that's white underneath? Okay, and so here's the two that we saw. This one is white underneath. and kind of has a very pale pinkish white spore print. And the one that's yellow-orange underneath has this yellow-orange spore print. And while we were doing that, we found another one that was pink underneath and had a pink spore print. And they were all in an area the size of this room. All three of these. So they're all together, they must be the same, right? So they're not. Okay, so we're describing, and this paper is in press at Mycologia, 3 new species, Chanterelle phasmatus, Chanterelle flavus, and Chanterelle spectaculus. Okay, and Roseocanus, we have more data in that paper about it, and I'll tell you more about that in a minute. So here's these two, the white one and the yellow one side by side. Okay, so they're really different. Okay, and so here's Chanterelle phasmatus. Phasmatus is, we call it the ghost chanterelle. Phasm is spirit or ghost. And it's white underneath when it's young especially. And there it is. It's very delicious. It's very, very fragrant. So we don't know for sure the distribution of it. We know it's outside of Wisconsin. We have markers from other eastern states. So we think it's very widespread, but we didn't, our intent was not to do a distributional study. So the Hymeneum is pale white to pinkish, and it has a pale or yellowish, or very pale yellow stalk, and the spore print is this kind of light pinkish white spore print. And there it is close up, very white underneath, but very yellow-orange on the top. And Chanterelle flavus, I couldn't believe this name wasn't taken, the yellow, eastern yellow chanterelle. Yellow spore print, yellow hymeneum, yellow stalk, everything about it is yellow, this kind of yellow-orange, yellow toward the orange side, not toward the green side. It's smaller, not as fragrant, but still it's good. Here's a closer view. This is a better color view of it. Very yellow, and sometimes clustered, sometimes not. Okay, this is Chanterelle spectaculus. This is the least common of our species by us, but it is yellow stalk, yellow cap, but the hymeneum is sort of this pink color, and so we thought that looked kind of spectacular. The spore print is salmon pink. So there's some examples of Chanterelle spectaculus, and there's Chanterelle phasmatus next to it for comparison. Okay, so these 3 things in an area the size of this room are 3 different species. It's a conspicuous species. It's not hiding anywhere. It's not hiding under logs. It's not hiding by being brown, being an Armillaria. They're edible. People know what they are, but we misnamed them. We're calling all 3 of these things the same thing, and we're calling it the same thing we called it in Europe. So, okay, so here's, I don't want to bore you with too much of this, but you can see that when we looked at the large subunit and just look at these things right here, we were able to separate Chanterelle spectaculus. Here's Roseocanus, and we were not able to separate the yellow one from the white one from this set of DNA. But we could separate it using other regions. And so we clearly could separate Chanterelle flavus from Chanterelle phasmatus. Here's Roseocanus, and the other one is in there somewhere. And this is a new region that Bart Buch has been pushing, the TEF1 region. And we have them pretty well separated now, Chanterelle flavus, Chanterelle phasmatus, Roseocanus, and spectaculus. And one of Bart's species seems to be the same, it goes in the same clade as one of our species. So the Chanterelle, one of his specimens of Chanterelle tenuithrix seems to be the same as one of our Chanterelle flavus. So that's still, we need more collecting to work out the total details of that, but for now they can be, there's enough data to consider them separate species. And so we're not sure of the range of all of these, and that distributional map will come later once people start to recognize the species. So it seems likely there are more species in the east than just this, that we would find 3 new species in an area size of this room, and that would be all there was in the whole east of the Rockies. Seems kind of shaky. And so more data needs to be collected for this. So it seems like all of these species that we're looking at so far have been very regional in their distribution. So we have some that are in the southeast, some that are just in California, some that are all along the west coast, up into Alaska. Some of them are in other places. Some of them are in deep south, some are in Gulf Coast. Okay? But Chanterelle roseocanus is different. And so this is the original site somewhere in there in Oregon. And other places we have DNA from are Idaho and Colorado. We have two different sites, two different areas of the state, totally different, that give us the same exact DNA sequence. And so, so we think that it's probably all down the Rocky Mountains. This seems to be the most common species, and I'll show you a picture in a second. We have data from just across the border in Michigan from Wisconsin. These are all under conifers, all under spruce mostly. We have data from upper New York State and from Massachusetts and Newfoundland. And so our hypothesis is that Chanterelle roseocanus is on conifers from here through the Rocky Mountains, and maybe we can follow it along the conifers up here and be across this whole northern conifer tier up and through this area. But maybe there's a disjunct population. We don't have any data. We're just guessing for now for a hypothesis. And so the very distinct species, bright yellow underneath, yellow spore print, and this is from the Colorado Rockies. This one's near in the western part of the state. And so the eastern ones look pretty much the same as this. Would you agree, Linnea? Yeah. Okay, and so they're quite good. I don't think they're quite as good as Chanterelle spasmodus, but I'm a little bit biased. They're quite good. I can't tell the difference too much between them. There's a lot that can be very abundant. So here's Kevin Bone with a whole bunch of them he was collecting. They tend to be much smaller than the original collections from Oregon, but they're apparently still the same. Here's a box from that collecting, beautiful things. Here they are frying in the pan. And here's one of Gina Bone's famous dishes from chanterelles and trout over potatoes. So very, very good. She's a great cook and has a nice book, so you should buy it for Christmas. And so we've learned something today. And I'm going to read you, this is the end, or in one of the last paragraphs of our Chanterelle paper that's in press. Molecular phylogeny has given mycologists the framework and competence to discern morphological differences that separate the species. Sometimes these differences turn out to be quite striking. What were we thinking? But often they are more subtle. Clearly, much more systematic work needs to be done on North American fungi, even the charismatic megamycota, to elucidate these cryptic species. Okay? So this says we need more work even on the stuff we think we already know, because we don't know it. So with that, I thank you very much. So I think we have time for a question or two if you got them. Yeah.
Well, we were calling, there's a chanterelle that's kind of shown up in the Salt Point, Sonoma Coast area that we've been calling rainbow chanterelle. But that's not the roseocanus?
Yeah.
It is the roseocanus.
Yeah.
Because I thought that was last one, the yellow was the roseocanus.
No.
Okay, thanks.
They're quite yellow, but they're different. You know, they can be different morphologically. Yeah. We don't see whitish in anywhere else but around here, around the coast.
And what about the Laetiporus, the smooth chanterelle?
That's a different species.
But is it still Laetiporus?
Yeah, because that was described from North Carolina. That's a North American name. Who's over here? Yeah.
How is it safe to separate these species based on their mycorrhizal patterns? For instance, up in Michigan, the normal chanterelles grow in cacti.
Yeah. So the question is, can we separate these based on their mycorrhizal partner? And the answer is yes, to the extent that we know them, but we don't know everything yet. So more study needs to be done based on that. So all of our species in Wisconsin are under oak. So different kinds of oaks. Yeah.
I just had a question on compatibility of studies.
not compatible because they're not the right mating strain?
That's a good question. So we don't do just one mating. We do a whole set of matings, and the chances that we would get just one mating type out of all of the spores we collected would be very small. And so what you're asking is that how do we know that we're just going to have the same mating type and they're not compatible? So we have different tester strains that we know are different mating types, and we test all of them against both of those tester strains, if you're doing it right. Anybody else? Okay, so if you have more questions, feel free to ask me, and thank you very much.